Okay, we're back, just beginning on Chapter 9, the nervous system. Before I start on the PowerPoints this morning, I want to give you a little preliminary information on the nervous system and Chapter 10, the senses. Tell you a little about the nervous system. This is off of some notes that I've had that I've gathered over the course of time here. The job of the nervous system is to gather information about, and I'm going to say, organisms, but it would be, in our case, the human organisms, internal and external environment. And then the organism or the nervous system would process and respond to the information that had been gathered. The information it gathers is in the form of electrochemical commands or signals, and these are sent to direct the appropriate response. We have a bit more information here on the senses. Rather on this nervous system in general first. The nervous system will regulate many of our simplest and most complex activities. The nerve impulses are generated and transmitted, again, electrochemically, and then they will regulate the functioning of tissue, of organs, and organ systems. And this allows us to perceive and respond to our external world as well as our internal environment. The detection now of changes such as I've just mentioned here is a function of our sense organs. Our senses, in Chapter 10 you'll see, this is still a study of the nervous system. And the functions now of our nervous system is guided, well, I don't know if it's guided or not, but it will detect changes and it will allow us to feel sensation. Secondly, it will initiate responses to appropriate changes. And thirdly, it will organize and store information. So, let's go ahead and begin Chapter 9. Nervous system is composed of neurons and neuroglia. Neurons, these will transmit these nerve impulses. These nerve impulses, again, are electrochemical signals, and these will be transmitted along nerve fibers to other neurons. A neuron and a nerve cell are synonymous. Typically, these nerve cells or the neurons will have a cell body, they'll have an axon, and they'll have dendrites. And I like to think of this as just the typical cell, typical nerve cell. It will have a cell body, as you see here, and then axons will extend out of the, you could say, the bottom of this cell body. It's like a long tail, and then it'll have a branching on the bottom of it that will help make connections to other axons or to organs, etc. Now, on the cell body, you're going to see extensions from it, and those will be the dendrites. So just so long as we know what the cell body, the axons, and the dendrites are. So nerves are made up of bundles of nerve fibers, and these nerve fibers then are the cell body, axons, and dendrites. Neuroglia, these carry out a variety of functions. They aid and protect components of our nervous system. Good illustration here. I want to really emphasize what I mentioned a while ago, the descriptiveness that I'd given. You can see the cell body there. You can see that that cell body does have a nucleus in it. That's that dark feature in the center. And you can see the dendrites extending from the cell body. Keep in mind, the dendrites will receive information from the environment, and it will transmit that into the cell body, and then that information is then transmitted through the axon to another nerve cell, or perhaps it would go to an organ, whatever it may be making connection with. We have organs of the cell body, organs of the nervous system can be divided into the central nervous system, the CNS, and that's made up of the brain and spinal cord. It is central, and we'll see an illustration here in a moment, just the brain and the spinal cord. And then we have the peripheral nervous system. I like to say the peripheral is that on the side. It's made up of peripheral nerves that connect to the CNS, that connect the CNS to the rest of the body. Here is a good illustration on slide 5 here in chapter 9. You can see the slide on the left there shows the brain, the spinal cord, and then that will be the CNS, the brain and the spinal cord. Then the peripheral system is going to be on the side, and that's going to involve the sensory division and the motor division here. You can see on the illustration there on the left, you can see all of the peripheral nerves, if you will, coming from that. The cranial nerves, spinal nerves, you can see how far they extend throughout our bodies. So the motor division now of this peripheral nervous system is going to be made up of the somatic nervous system and the autonomic nervous system. Somatic nervous system, that of the body, is going to be primarily skeletal muscle. Keep in mind, skeletal muscle moves our skeleton. Then we have the autonomic nervous system that we cannot control. The somatic nervous system, we can control it. We can tell our bodies what to do with this skeletal muscle. But the autonomic nervous system, that is smooth muscle that moves product through our digestive system, that allows our retina to open and close according to the light intensity. It has to do with our cardiac muscle, the beating of our heart, etc., and our glands as well. All right, functions of the nervous system. I've given you an introductory statement about that, and I still like that. This says that there are sensory receptors at the end of peripheral nerves that gather information and convert it into nerve impulses. Keep in mind, this is electrochemical impulses. When the sensory impulses, these electrochemical impulses, are integrated in the brain as perceptions, we term that being integrative. It's the integrative function of the nervous system. We have conscious or subconscious decisions that will lead then to motor functions via certain effectors. So I put my hand on the stove and I determine that it's hot. So I made a conscious decision here, take it off. The motor function comes through the effectors. The effectors there will be my muscle system of the somatic system. So motor functions can be divided into the consciously controlled somatic nervous system and the unconscious controlled autonomic system. The autonomic system is, like I described before, would be like moving product through our digestive system or our retina pupil there opening and closing. Talk about neuroglia. Neuroglia cells. These fill spaces They support neurons. They provide structural frameworks. They produce myelin, and they carry on phagocytosis. All right, myelin. These are wrappings. This is like a layer of fat, so to speak. It's wrapped around, a lot of times, these neuroglial cells. We have microglial cells that are small, and these will be phagocytic, and they will clean up bacteria and other cellular debris. We also have oligodendrocytes. These are the things that form the myelin in our brain and in our spinal cord. Astrocytes will be star-shaped, so to speak. They are near blood vessels. They support structures, and they assist in metabolism, respond to brain injury. They will fill in spaces. Fourth bullet there on slide 8 is the epidermal cells. These cover the inside of the ventricles. This is ventricles in the brain, and they form these cord plexuses within the ventricles of the brain. And then we have Schwann cells. These are the real producing myelin neuroglia of, note here, of the peripheral nervous system. Schwann cells. Good illustrations right here. I want to emphasize to you, you can see the neuron there on the upper right. You can see how it connects over there to the latter appearance there of the oligodendrocyte. You can see the microglial cell there. You can see the astrocyte, the star-shaped feature on the left. You can see even the circulatory system being noted there as a capillary in pink. This is on slide 9. If you will, look in the center, lower center of this slide, and you're going to see the myelin sheath being cut, and you can see the layering of it. This is laminated. You'll see in a little bit that it talks about lamellae. These are the laminations of this myelin sheath, and it helps to insulate, if you will. It acts like insulation on a wire. Now, right below that exposed myelin sheath, you see a little feature that's got a bracket around it that says node. Those are the nodes of Ranvier. And the electrochemical impulse will jump from node to node. You can see a node above that one and a node right below that one. So it jumps from there to there, continues on down the axon. So we have these. Let's go on to the next slide. This is a real good slide. Oh, one thing I didn't point out there on that slide 9, the epidermal cell. You can see those that surround features there like the brain or in the spinal cord up there. Okay, we said before that a neuron contains a cell body. It has a tubular cytoplasm that it has these dendrites that will extend from the cell body, and it also has an axon, as I described earlier. The cell body contains, as does many of our cells, mitochondria, the powerhouse of the cell. It also has lysosomes, it has a Golgi apparatus, it has nysyl bodies, and it has rough ER, rough endoplasmic reticulum, as well as neurofibrils. Good illustration here. You can see all of the features that I've already mentioned there. You can see the nodes of Ranvier. You can see the myelin, that sheath that is wrapped around the axon. One thing I want to note back up toward the cell body there is a axonal hillock. This is what will be the beginning of the axon. So pay close attention to that, and notice that is all of this through the axon there is made up of neurofibrils. We have branching dendrites that carry impulses from other neurons or even from receptors, and it carries them toward the cell body. The axon then will transmit the impulse away from the axon through the axonal hillock of the cell body, and it may have different side branches that it would extend off of the axon as well. Larger axons, it says here, are enclosed by myelin sheaths. These are provided by Schwann cells, and these are myelinated fibers. Outer layer of the myelin is surrounded by a neurolemia. That's that wrapping that I mentioned. It's made up of cytoplasm, has nuclei of the Schwann cell. And I'd mentioned the nodes of Ranvier there, the narrow gaps in the myelin sheath between the Schwann cells being called the nodes of Ranvier. Good illustration here again on slide 14. Notice here that this is a Schwann cell, these laminations, these myelin sheaths. It is a cell, and we know that it's a cell because it has a nucleus in it. You can see that blue feature there where it's showing the Schwann cell, as well as in the, you can see a piece of it there in the little square that is pulled out at the bottom. Alright, the smallest axons lack a myelin sheath, and thus we say they are unmyelinated fibers. White matter in the central system is due to the myelin sheaths in this area. Unmyelinated nerve tissue in the CNS makes up what we know of as gray matter. So white matter and gray matter in the CNS, in the central nervous system. What is the central nervous system again? It's the brain and it's the spinal cord. Now, let's go on to the peripheral system. These peripheral neurons are able to regenerate because of the neurolemia. And the central nervous system axons are myelinated by these oligodendrocytes. Thus, they lack neurolemia and do not usually regenerate. Neurons can be grouped in two ways. On the basis of their structural differences, they'll be bipolar, unipolar, and multipolar. We're going to see illustrations of these just in a moment. I'm on slide 16. And by functional differences, they'll either be sensory neurons, they'll be interneurons, or they'll be motor neurons. Let's find out a little bit more about the multipolar and bipolar and unipolar. Multipolar have many nerve fibers that arise from their cell bodies and are commonly found in the brain and the spinal cord. Brain and spinal cord, multipolar neurons. Bipolar, don't go there about being bipolar. It's totally different. Bipolar neurons are found in the eyes, nose, and ears, and they have a single axon and a single dendrite extending from opposite sides of the cell body. We're going to see some illustrations of these right here. Multipolar, you can see the direction of the impulse. You can see the dendrites going through the cell body. They would receive information there from the dendrites, and the information, electrochemical information, would be transmitted through the cell body and on through the axon, be carried to another nerve cell a little farther down the line. You can see the bipolar. Notice here that the dendrites will carry the information again to the cell body and then on through the axon on to another cell. And then the unipolar, you can see the direction of impulse there again. They're on the peripheral processes, and it will go on through the axon and the cell body and make its way to the next cell. Okay, the unipolar ones are found in the ganglia outside the CNS, and they have an axon and a dendrite. And notice, if you will, on the illustration again, that cell body will arise from that little short fiber there on the axon. All right, and let's talk about sensory or afferent and efferent neurons for a moment. Sensory neurons are also termed afferent, going toward. These conduct impulses from the peripheral receptors toward the CNS, central nervous system. And these are usually unipolar, but some of them may be bipolar. The interneurons are multipolar. They lie within the CNS. They form links between other neurons. Cell bodies here some interneurons will aggregate in special masses called nuclei. These motor neurons are multipolar. These conduct impulses from the CNS to the effectors. In other words, it goes from our central system, our brain, through our spinal cord, out to the effector, and the effector might be a muscle, a gland, etc. All right, a synapse is simply the space between two neurons that would be communicating with one another. This space, also termed a synaptic cleft. You have a presynaptic neuron, you have a postsynaptic neuron, and the synaptic transmission is where it crosses the synaptic, is a process where it crosses the synaptic cleft. Okay, I've mentioned electrochemical features within our system that conduct these messages. These are termed neurotransmitters. They are biochemicals. They carry out synaptic transmission process within the synaptic cleft. You have synaptic knobs and synaptic vesicles. Now these synaptic, this is on the distal ends of the axon, on the long tail. Go back and look again at your illustrations. You have synaptic knobs. These would be like at the tips of your fingers, if you want to say that. And within the tips of the fingers here, those knobs would be synaptic vesicles. A vesicle holds something, like a vessel holds something. Here is where we'll find the neurotransmitters. The action of these neurotransmitters can either be excitatory or inhibitory. Real good illustration here. You can see the synaptic knob there. You can see the little blue dots inside there being the synaptic, being the neurotransmitters, and the little encasement there being the vesicles. You can see how they're released in that bottom portion of the slide 22. They're released into that synaptic cleft, if you will. And notice where they go. They go across to those little features that kind of look like cups. They are those receptors on the other side, on the postsynaptic side of the synaptic cleft there. So that's how that will make that connection. All right, cell membrane potential. A cell membrane is usually polarized, in other words, positively charged or negatively charged. Usually these have an excess of negative charges on the inside of the membrane. And in order for conduction of nerve impulses to take place, polarization has to be in place. All right. Here you can see again the phospholipid bilayer surface that we keep mentioning, that is the cell membrane. You can see the fatty acid tails on the inside of the phosphate heads there. And notice that embedded in this phospholipid bilayer surface is what is a protein of types. And it has a gate-like mechanism as it's described. You can see there in slide A that it's closed. In bullet B there, the channel then is open. All right, we're talking here about distributing or distribution of ions. Again, you've heard me say this numerous times, ions are charged particles. And these will be positively charged or negatively charged. The distribution of these ions is going to be determined by the membrane channel proteins that would be selective for certain ions. So we saw a good illustration of that on the phospholipid bilayer surface there a moment ago. We'll talk about potassium and sodium here as well. Potassium ions will pass through the membrane easier, more readily than do sodium ions, making potassium ions a major contributor to membrane polarization. There's a feature that we term resting potential. This has to do with active transport. Active transport. We'll find out a little more about this. Requires energy. The cell maintains a great concentration here of sodium ions outside and a greater concentration of potassium ions inside the membrane. The inside of the membrane has excess negative charges while the outside has more positive charges. This separation of charge, called a potential difference, is the resting potential. And you have good illustrations there. You can see in the lower nerve cell there, you can see the red features there. It is talking there about the sodium-potassium pump. And I encourage you to read each one of the bullets, A, B, and C, and that's going to give you a little bit better information than what I can provide for you here in this lecture. All right. Potential changes. Stimulation of a membrane can locally affect its resting potential. Keep in mind, resting potential. When the membrane potential becomes less negative, we say that the membrane is depolarized. It's less negative. If it's less negative, what else do we know? If sufficient strong depolarization occurs, there's a threshold potential. We say threshold is achieved or is reached as the ion channels will open. At threshold, action potential is reached. Action potential then may be reached when a series of sub-threshold stimuli summate, come together in order to reach threshold. There has to be adequate positive and or negative charges in place for action potential to be reached or threshold to be reached. Action potential. At threshold potential, I'm on slide 30 now, membrane permeability to sodium suddenly changes in the region of stimulation. As the sodium channels open, the ions, the charged particles now, rush in and the membrane potential changes and it becomes depolarized. And at the same time, you'll see this in a moment, at the same time the potassium channels open that allow potassium ions to leave the cell and the membrane becomes repolarized. So there's this constant cycle of polarization, depolarization, repolarization, polarization, depolarization, etc. Rapid sequence here is the action potential. Active transport mechanism, of course, is going to work in order to maintain the original concentrations of sodium and potassium. Good illustrations here again. You can see the dots representing sodium and potassium. The yellow will be the sodium. The blue ones, purple ones there will be the potassium. Both of these are positively charged. No, I'm sorry. I've confused you. The sodiums are negatively charged, the potassiums are positively charged. So you can see this cycle through here as to what is taking place. And I'll back up on that. Sodium and potassium both are positively charged. All right. What is a nerve impulse? It's conducted as an action potential and it's reached at the trigger zone. This spreads by a local current flowing down the fiber and adjacent areas of the membrane reach action potential, as you can see taking place here. All right. Unmyelinated fibers. These have impulses that cover the entire membrane surface. And myelinated fibers conduct impulses from one node of Ranvier to the next node of Ranvier. This is called saltatory conduction. Saltatory conduction is very fast, faster than it would be on unmyelinated neurons. The all or none response: if a nerve fiber responds at all to a stimulus, it responds all together. We term this all or none response. There's greater intensity of stimulation. If there is greater intensity of stimulation, it triggers more impulses per second, not stronger impulses. More impulses per second. So we know that it is stimulated. Okay, I'm going to stop right here and we'll take a break.